So basically, bioinformatics is the field that analyzes biological data, and this includes genomics, proteomics, systems biology, like text mining, anything that is related to biology, basically. And there are two main applications of bioinformatics in industry, and that's in precision medicine and automated diagnostic tech. So I know that I just threw like a lot of vocab words at you guys in like 30 seconds, but don't worry because we'll be unpacking each of these terms as we go through this overview. So let's look at how bioinformatics can be applied to genomics. So genomics is basically the structure, the function, basically anything that has to do with the genome. And the genome is the complete set of our DNA. So all like 10 billion bases. And the application of bioinformatics to genomics is that you can develop machine learning algorithms that can automatically determine the location of protein encoding genes in our DNA. So this is known as gene prediction. And you guys may be wondering, well, didn't the Human Genome Project already like sequence the entire human genome like way back in 2001? Yes, they did, but we still don't know the function of most of these genes, and we don't know which ones are protein encoding and which ones aren't. And figuring out which ones are, which ones aren't, could really help us in understanding the process of aging, maybe even find potential treatments to cancer, and even find treatments to like inherited diseases and everything. So this is a really important field, but it's just so tedious to go through like our entire genome and try to find the structure and the function of every single gene and what proteins it produces. So, and this is called DNA sequencing. And so basically we're using AI to try to locate regions in the DNA that encode proteins and not doing it by hand. So basically how this works is DNA contains introns and exons. And exons are regions of your DNA that are protein encoding. So they, like I said, they code proteins that may have to do something with cancer, that may contribute to aging, that may prevent aging from happening, like all that. And introns are regions that aren't protein encoding. So these, while in the past, these were thought to like be like useless regions, researchers are now finding out that they're also contributing to like regulatory functions. And so those are also important, but that's not the focus of like DNA sequencing at the moment. Right now, we're just trying to find protein encoding regions. And locating these regions can be done through two ways, intrinsic and extrinsic searches. I'm having some trouble. Okay, okay, cool. Oh. Okay. So an extrinsic search is basically the tedious like manpower method where like brute force method where you basically search the target genome for sequences that are similar to a known protein encoding sequence because you all like the DNA is basically just like this repeating list of 4 letters. And so you can search this list of like 3 billion base pairs for sequences that are similar to ones that we know encode proteins. However, if you do this by hand, it's really expensive, they're difficult to find, and you're really prone to missing potential protein encoding sequences, and you're also prone to error. So that's why we're trying to use machine learning to create gene prediction algorithms that can use machine learning to identify segments of the genome that could potentially be protein encoding. And then these segments can then go through like the analysis of a bioinformatics researcher or structural biologist and actually figure out who can then figure out whether those regions are actually protein encoding or they aren't. So basically really shortens the time taken to find these protein encoding sequences. And it basically so it streamlines it. And certain machine learning and deep learning techniques that can be used in this intrinsic search are random forest, k-nearest neighbor, support vector machines, and multi-layer perceptrons, which are basically simple neural networks. So the reason that there are so many techniques listed here is because researchers are always coming up with new ways to solve this problem from like a different angle. And we'll also be learning at the end of this workshop how to apply some of these techniques like random forest, for example, to our own data sets. So that'll be exciting. Okay, so machine learning can also be applied to proteomics. And just like with genomics, proteomics is basically anything that has to do with our proteome, which is all the proteins that are produced by our body. And proteomics is a really important field because proteins are responsible for so many different biological functions. They're responsible for the production of ATP in cellular respiration. They are responsible for making our eye clear, and like there's digestion proteins, they just have so many different functions. But while we do know the functions of a lot of proteins, there are also a ton of proteins that we don't know the functions of, and we're discovering new proteins every day. But we do know this, that protein folding determines the function. And protein folding basically occurs in 4 stages. There's the primary structure, the secondary, the tertiary, and the quaternary. The primary structure is basically just the string of amino acids, so that doesn't really give us much to work off of. But the secondary structure is when the protein first starts forming its interactions, and that determines the preceding structure, so the tertiary and the quaternary. So we also know that the structure of a protein can help us predict its function. So proteins with similar structures can be grouped together into similar groups, and they might have similar functions. And so we can apply machine learning by predicting the secondary structure of a protein. And we can use stuff like deep convolutional neural fields to do that. So in addition to finding the function of proteins by predicting their structures, and just a side note here, a company came out with this algorithm called AlphaFold, which is the first like commercialized algorithm that can actually accurately predict the structures of proteins. And this raised like a huge debate about whether this would remove structural biologists from their jobs, and whether that like AlphaFold was threatening the jobs of structural biologists. So I'd like to pause here and maybe ask for your guys' input. What do you guys think about AlphaFold and its impact on this job that was previously seen to be pretty secure? You guys can feel free to jump in.
If I may attempt to respond to that, I mean, I do feel like that protein structure prediction is very complex. And what AlphaFold has achieved is pretty incredible by the standards that we have now. But I do feel like, you know, it's the same thing that if you could make an analogy to the Human Genome Project, when Human Genome Project was going on, people could be seen making claims like, you know, cancer is going to be the thing of the past and stuff like that. And like, you know, 20 years later, maybe we know more types of cancer than we did like 20 years ago, right? When the Human Genome Project. So it may be the same kind of story may unfold with this structure prediction problem, because there is so many layers of complexity that, you know, we're going to need a lot more advancement. So it does work pretty well with the kind of data sets that we have. But I feel like there is a lot more to the problem. And as soon as we discover new proteins and new function, then if we are to move towards synthetic proteins, you know, for which there are no homologs in the nature, we are going to run into a few more issues. So it's not really the end of the story, but that's my take on it.
Yeah, that's a really good point.
So hello. Just a few comments. I equally am of the opinion that AlphaFold isn't the end of the story for a few reasons. One is protein folding it's quite a complex process which hasn't been fully, elucidated beyond what can probably be programmed into any machine. There are various structures, structural components, other proteins and other structures within the cell, which facilitates its folding, which means there could be errors in folding. I mean, we have such example as misfolded proteins, prions, and the likes. So this just highlights the fact that protein folding, there are probably biological factors inherently, which perhaps might not be able to be programmed explicitly into a machine or far beyond what an algorithm could approximate, such as deep learning. Then moreover, so that would be the first reason. Another is due to the fact that AlphaFold even doesn't, it isn't 100% accurate. Majority of the cases which were solved by AlphaFold in the recent protein folding contest were sort of the simpler types of proteins, not the complex aggregates. I mean, not monomers, as it's performed quite well on. Or when it comes to complex proteins, which constitutes a bulk of what we see in biological systems, it fails at it. As a matter of fact, it's failed at numerous examples of this type. So it's a great leap. However, one would still be quick to highlight the fact that we still need human agents to still perform the usual experimental procedure. It's good at best when it serves as a form of augmentation to lab scientists. We would still need humans very much around. It's far from the end of the story.
Yeah, that's a really good point. So basically what I'm hearing is AlphaFold is not the end of the story, and we still need the input of structural biologists as you know, so that AI algorithms can get more advanced. So yeah, and another application of AI to proteomics is in protein constitutions. And this is a bit similar to protein structure prediction in that we're trying to classify protein patterns. So basically, instead of trying to predict the structure, we're just trying to group proteins into groups based on their patterns. And the reason this is important is because, as I said, protein patterns, or like proteins with similar structures have similar functions. So if we can group them together based on their pattern or their function or their structure, then we can also understand a bit more about how they're interrelated and what their possible functions could be. So how this works is images of proteins are generated using high throughput microscopy, but they're generated so fast that it's not possible for a human to sit through and classify each and every single one of the proteins in those images. So that's why we're trying to develop AI algorithms such as deep convolutional neural fields that can be used to classify the proteins in these images. You can also use machine learning in gene expression analysis. So gene expression is measured using a DNA microarray, which is basically kind of like an assay. And gene expression is basically the rate at which DNA is converted into instructions for making a protein. So gene expression can help identify tumors at a molecular level. And this is really important because in underserved countries or areas basically in which there isn't easy access to both diagnostic and like treatment healthcare, it's really important to catch cancer at an early stage because oftentimes the people who live in these areas might not have the resources or even the time to get to areas like metropolitan, or what's it called, like more areas with more infrastructure and more resources to treat higher stages of cancer. So, but it's kind of like a catch-22 because if you live in that area, you also probably don't have the resources to catch the cancer early. So that's why DNA microarrays are really important in that you can identify the tumor at a molecular level. So you can catch it really quickly, like before you even begin showing symptoms. And this can help you get treatment earlier, especially if you live in that area. So people are trying to use machine learning to read these DNA microarrays and identify which genes are being expressed. So for example, you can use AI to detect whether the proto-oncogene is being suppressed and whether the oncogene is being like overexpressed. And this can help you determine that, oh, like this person probably has cancer. And then that person would go to a medical center to get treatment. And their chances of survival would greatly increase. So some algorithms that can be used to analyze gene expression are naive Bayes, random forest, and QGIS. My computer is frozen. Okay, so this is kind of more of a niche application of AI, but stroke diagnosis and artificial intelligence are two fields that go really well together in that you can use AI to analyze neuroimaging data and predict stroke. And a lot of researchers have done this, even a few companies to varying levels of success. Most recently, there was a company that was able to get FDA approval for their algorithm to detect stroke on stroke images. So, or on neuro images. So that was something that's really interesting because as we all know, or it's a common saying in the radiology community, or I think it's a neurology community, that time is brain when it comes to stroke. So this is because for every minute that a stroke is left untreated, 2 billion brain cells die. And as we all know, after a certain number of brain cells die, the brain is basically dead. So we want to try to minimize the amount of brain cells that die, because even if like 50 billion die and the person still gets saved, they could suffer from long-term permanent brain damage. So basically, the quicker we treat the stroke, the better. But a lot of times, people don't really detect strokes in time, maybe because they miss it, because they're in a rush, or because the patient just has to wait behind like 50 other people to even get diagnosed. So what AI algorithms can do is they can run these scans through the algorithms, and the algorithm can basically predict which ones have a stroke, put those at the front of the line so that the doctor can see them first, confirm they have a stroke, and then treat them. So this is really important to minimizing treatment time and really maximizing patient outcomes. And you can use convolutional neural networks and support vector machines for stroke diagnosis. So another application of AI to biology is in text mining. And this isn't really the, it's not really like a conventional application because like obviously like text mining doesn't really have anything to do with biology, but it's still really important because currently thousands and thousands of research papers are being published about like every field imaginable in biology. So for a researcher or a professional to stay up to date on the literature in their field, they would have to read hours and hours of research papers. And oftentimes the research papers talk about really similar topics. So they're basically like, if they just read one of those research papers, they would have gotten the information from like 10 of them. So it's a lot of time that's being spent slogging through all these research papers. And it's basically impossible to stay at the forefront of your field because you wouldn't like even be able to work on your field. You would be like, reading research papers all day. So that's why researchers are trying to use text mining, which is where they create, they use natural language processing to extract relevant information from thousands of research papers and consolidate it into like this much less time-consuming report that researchers can read and stay up to date on what's happening. So this is really important in fields like coronavirus research and basically any medical field because doctors always need to know what's going on at the forefront of the fields. So now that we have an overview of different areas of biology in which we can apply machine learning, and you guys kind of maybe have some ideas of ways in which you can do individual projects, we're going to go through some sample bioinformatics projects in which you can actually put this this skill of AI to use in the field of biology. And you can actually figure out, like, maybe if you have an idea about a microarray, this may maybe help you figure out like exactly what steps to take to complete that project. So before we do that, I'm just going to go through a couple, the theory behind a couple AI algorithms that we'll be using so that you understand how they work when you use them. So the first algorithm that we'll be using is called random forest. And what random forest does is it differentiates your data by differences between the values. And this can be categorical differences, or they can also be numerical differences. So like if your value is a bunch of numbers, or if your data is a bunch of numbers, you can also use random forest. Similar to if it's a bunch of categories, you can also use random forest. And by differentiating the data, it turns it into a tree. And this tree can use a dichotomous key type classifier to classify the data into different categories. And it's called random forest because there's not like one single tree. There are a ton of different trees, and their outcomes are averaged for the final decision to try to get the most accurate decision. So here's an example of a dichotomous key, and this is kind of like what one tree in a random forest would look like. So in this example, the data is differentiated by categorical values. So for example, has fur or no fur, has feather or no feathers, and so on. So basically the machine learning classifier could go through this dichotomous key type thing and like classify it this way, and it would get most of the times the correct answer or the correct category. So coding the random forest is really easy with scikit-learn, which is a Python library. You basically just have to write 2 lines of code. So you call the model by naming it. So I'm naming my classifier, and then this is the random forest classifier function, which is imported from scikit-learn. And then n estimators is the number of trees in my forest. 100 is kind of a high number. It would take a pretty long time to train that. So I would say for the purposes of this workshop, you guys should keep it to 50 or below. And more trees can mean higher accuracy, but it can also mean overfitting. So let's just pause here, and can somebody like, so I'm not like talking too much, can somebody maybe like explain what overfitting is? If they want to, in the chat.
Sure,
Or
I can explain it. Overfitting is when you use a model, or are you training with data that does not have enough variation in it so that it fits real well with the training data. But when you go to use it with your test or actual data, you're going to have a lower percentage of accuracy.
yeah, that's exactly right. So having too many trees can cause overfitting by basically if the data runs through the model so many times, it would basically memorize like the parameters needed to produce those results, and that wouldn't be it wouldn't be able to generalize to other data sets. And then in terms of criteria equals entropy, there's a pretty long explanation for that. So I'm going to skip that for now, but if anybody has questions at the end, we can go back over why one of the criteria is entropy. And random state equals 2 is just what I found through trial and error works best. I don't really have justification for that, but it works. So yeah. And then how you would fit the model, because you basically created the model, but you haven't fit it to your training data. So it's not going to be very accurate right now. So by fitting the model, I'm basically saying how we're going to train the model. And you would train it by just saying classifier.fit, because there's a .fit function that you can import from scikit-learn. And then this is just my, the labels of my, or sorry, the data and the labels of my training data. So stats and diagnoses. And that's basically it. You're done coding your random forest in 2 lines. So the second algorithm that I'd like to go over is called naive Bayes. And in a nutshell, it's basically the probability of A happening given that B already happened. So this is used a lot of times in email spam classification or email spam detection. So if we think about it this way, like if you have 100 emails in your mailbox and you're trying, and the probability of an email being spam from these 100 emails is like 2, like say 2 of those emails are spam. So the probability of you picking a spam email is 2%, right? 2 out of 100. But if we use naive Bayes and we say the probability of an email being spam given that the email has the word deal in the subject line, say only 10 emails have the word deal in the subject line, and out of those 2 are spam, then your probability of picking a spam email from this smaller subgroup rises to 20%. And so you're going to be right more percentage of the time, like a greater percentage of the time. And that's basically how naive Bayes works. It makes it easier to guess the correct answer. Does that make sense? Okay, perfect. So this is the formula written out for naive Bayes, but we don't need to worry about that. We will not be coding that because we have scikit-learn. And scikit-learn takes care of all of the back-end mathematics for us. And like we had to do with random forest, we only have to write 2 lines of code, which is amazing. So we basically call the model just by calling Gaussian ND. And we don't, that function doesn't take any parameters. So we just have empty parentheses. And then we just fit the model to our training data. And that's basically it. So now that we have an overview of different ways in which bioinformatics can be, or AI can be applied to biology, and also the theory behind different AI algorithms, I thought it would be nice to maybe go through some case studies of how we would actually go through and code those algorithms and apply them to actual data sets. So before me, I have 2 data sets. One is a diabetes data set taken from UC Irvine, I think. And we will be trying to predict based on different factors of a person's like biographical data, whether they will develop diabetes or not. And then the second data set, the one on the right, is the heart disease data set. So we will also be predicting based on different factors, whether a person has heart disease or not. And yeah, that's pretty much it. So, I mean, you guys tell me, do you guys want to go through both of these as a group, or do you want to maybe take like 15, 20 minutes to make a copy of these notebooks, try to work through them by yourselves, like one of them, and then if you guys have any questions, we can like have like an open forum, or anything is fine. What do you guys want to do? As a group, okay. Yeah, I'm gonna send the links shortly. So for the diabetes data set, this is a data set that I'm sending right now. And please make a copy of the notebook, the Google Colab notebook, because this is like a centralized copy that everyone has to use. So
So as you know, Zoe, I'm one of the complete novices on this. So once we've got the data, it would be great to sort of.
yeah. So basically, here I can just go through, or since we're going through it together, I can just tell you guys like how I would do this. So starting with the diabetes data set, I'm just going to click on this. And this takes me to Kaggle, which is this really great community for people who want to learn data science. They have like competitions, really clean data sets, so not much pre-processing needed. And yeah, just a lot of really great stuff. And you need to sign in to or register to create or to download data sets. And they don't like spam or anything. So I would say if you're interested in learning more, it's a good idea to register for an account. So I'm just going to sign in with my account. And I'm going to pause screen sharing while I do that. Okay, So now that I'm signed into my account, I'm just going to download. I can see this button over here, so I'm just going to download. Oh wait, you guys can't see. Can you see now? Okay, I'm assuming you guys can see.
Yes.
Yeah, we can see now.
Cool. So I'm just going to download the data set. And yes, I went to my downloads. And then I don't know like how it looks on your guys' computer, but on Safari and like the one from like 10 years ago, because this is an old computer, I'm going to click, or no. So it's been downloaded, I can see. And then I'm going to go to the collab and have it open. Okay, so unfortunately, this opened with Microsoft Excel, which is not what I was trying to do. So I'm going to close that. Okay, so what I'm going to do is I'm going to make a copy first. And to make a copy, you basically just save a copy in Drive. And I'm going to open into tab. Okay, and then I have this file and I want to use it, but like, where do you upload the file? So basically on the left-hand side, you see like these 4 icons. You can upload it to this folder looking thing and it's connecting. Okay, so then I basically click like this little page thing and this allows me to upload files to my runtime. So I'm gonna stop sharing while I upload the file. I need to find it. Okay, so I found it and mine is called diabetes.csv. And as long as you guys like didn't change the name of yours, it should be fine. Like it should be the same as mine. So then I just press like upload and it says the files will like get discarded when the runtime is recycled. So just press OK to that. And then your file basically gets uploaded. So now that I have the file on my runtime, I can basically run this block of code. And this is where I import all of my libraries from Python. And like do you guys remember when I said like scikit-learn makes everything super easy? So this is scikit-learn right here. And this makes everything really, really easy to train and visualize and everything. So yeah. And then over here is where I'm loading my data frame. And I'm doing this with the help of pandas, which is another super, super helpful Python library. And whatever you guys have named your file, I named mine diabetes.csv, as you can see over here. So whatever you've named it is what would go here. And when you run it, you should be getting like a really nicely formatted table of this data set. And as you can see, there are a couple of parameters that the people who created this data set recorded. So they recorded how many pregnancies a person has, what their glucose level is, what their blood pressure is, their skin thickness, their insulin level, their BMI, something called the diabetes pedigree function, their age, and their outcome. So outcome is basically the label. That's what we're trying to predict. 1 means they have diabetes and 0 means they don't. So does this all make sense so far? Okay, perfect. So now what I'm trying to do is I'm just going to get this data from the inside of the table and make it into something that I can upload to my machine learning model. So that's what I'm doing over here. And then basically, so this is like my training data, like my training parameters. And then over here in these 2 lines, I'm creating my training labels. Or not my training, but you know what I mean. I'm basically separating the data into like the parameters. So all of this stuff and then my labels. So just the outcome. And that's what this is doing. And then over here, I'm using scikit-learn, the train test split function from scikit-learn to split my data into a training set and a testing set is what this is doing. And then over here, I'm just scaling my data because as you guys can see, it goes anywhere from like 0 to 148. And this would A, take a really long time to train, and B, it wouldn't be very accurate with such a wide range of data. So I'm just going to scale it down and as you can see now, it is a much smaller range of numbers, so the decision tree can train faster. And then over here, I am calling my classifier, so random forest classifier, all that stuff. That should be it. And then over here, I'm fitting my classifier to the training data, and this is just basically where it trains. And then over here is just a small function that I created to kind of show the actual value and the predicted value. And as you can see, it did pretty well. And over here, well, like over here, I can only see like the first few values. So yeah, and let me just stop there. So basically what we've done now is we have created our data set, split it into training and testing data sets, created our machine learning model, random forest, trained it, and then tested it as well. And now we're looking at our testing data and what the results were. So is everybody with me so far? Or am I going too fast or anything?
You might be going a bit quick.
Okay. Is there anything that somebody's confused about that maybe you want me to go over again? Or just like any questions at all?
Yeah, can you just and if I'm the one who's the slowest on here, other people please say yeah, okay, it's not just me then. So if you can go back to the top, can you just go through each programming and show us? So I understand that we're reading the file diabetes CSV, and then we're at the next one that talks about the columns. So can you just walk us through each one in English in terms you've got pregnancy and age? And I know that you did it, but you did it a bit quickly for me.
Oh, sure. Yeah. So basically the goal of this like cell block here and this cell block here is to separate this nice looking table into two different like files kind of. One that would basically be like the parameters. So like all the inputs basically. So like pregnancies, glucose, all that stuff, like everything that affects whether you get diabetes or not. And then one would just be this column of the corresponding outcomes to all these inputs. So does that make sense?
Yes, and so you've got pregnancy and age because that's the from and the to and everything in between.
Yeah, like this is my first column, age is my last column, and then I just create a table called stats where I go from the first column to the last column plus one because it is not inclusive at the end.
Sorry, plus one because
it's not inclusive. So I need to add one to make sure I don't cut off before the first column or the last column.
okay, that's helpful, thank you.
Yeah, and then I want the values from that, so that's why I did dot values, and then I just printed it to make sure I got it, and it looks pretty good to me.
I've got a question, if I may. Do you know how big that whole data set is? Because obviously you're only printing out the first few lines of it. How big's the whole thing?
Oh, I don't know. I think we can look here though.
Yeah, I tried looking through there. I didn't see it obviously. It might be a couple of hundred.
Yeah, it looks like it's a couple hundred rows.
But it,
I think maybe you could do like, what is it called? Like,
ALOC has said it's 768.
oh, cool.
that's better.
Yeah. And then over here, we're doing the same thing, but we only want one column. So that's why I'm just going from this column plus one, dot values, and I would print it if I wanted to. Yeah, you could do like len df or like df dot. I think size would give you like the size of the matrix, but you could use that to find the values as well. Okay, so is everyone good with how I separated it into the inputs and the outputs now?
So I understood from the pregnancy to the age, the second bit of coding, the outcome. What is the purpose of this?
So I basically created a matrix with all of my inputs, but I also want like a list or like a matrix with all of my outputs that correspond to these inputs, because we need to give our training data like a label of what it should be getting based on those values, right?
Understood.
Yeah. So That's what this does. It basically creates another matrix that is only the outcome that correspond to the inputs in this matrix that I created here.
Okay, great.
Okay, and then over here is where I'm splitting it into stats train, stats test, outcome test, and the test size is 25%. So I think I had a question in the chat. And by the way, I'm sorry that my chat is like not working, but it's like not notifying me. But the train test split is 75-25. So the testing size is 25% and the training size is 75%. And I think the standard is 30-70, but I was just like playing around with different numbers. You guys are free to do like 20-80, 25-75, 30-70 is, like I said, standard. I would say don't like do 50-50 because that would be a very large testing set. But yeah. So is everyone good?
Yes, I have just one question. Is it possible that we can somehow define our own training set? Then we can add new like test or whatever.
So like, are you saying set manually?
Yeah. So let's say that I have a small data set, and I want to actually define the training set. If there's an option to do that.
Yeah, you can manually create your training set, or you can like create a function to create your training set. But I would warn against doing it like very manually. I would say create a function that randomizes the process, just to like preserve like the scientific integrity of it and everything. And also it would be like really tedious and labor intensive to create a training set for very large data sets. But I think I heard you say that like if yours is small, if you wanted to do it, yes, you could. But I think it would be better to do it randomly. And if you don't want to use trade test split, the reason I use it here is because it's like one line. But if you don't want to use it, like if it doesn't fit to your data or something like that, then you can always create like a function that does it. Like you can create like a bespoke function basically that does it manually, and that fits to your data. Does that answer your question?
Yeah, more or less. Thank you.
Maybe I've understood the question slightly different. Were you asking about how to get your own file with the data that might be in a different format in there? Is that?
No, I was trying to ask whether we actually can, let's say, pick the samples that we can classify them as the training set. That's it.
Ah, okay. So not a different data set, but specify which parts are training and which parts are test. You really,
Yes.
you don't want to be doing that. That's a bad idea. I think that is what Julia was trying to explain.
Yeah, you should
Why that's a bad idea.
You should try to make it as random as possible to preserve the scientific integrity of it, because you don't want to go publishing research where you've like hand selected the data.
In fact, it's very, very common practice to try multiple random splits. Do it again, but with a different random split, and see if the results are consistent.
Yeah.
And by doing it non-random is what leads to the overfitting, can lead to overfitting.
Yeah.
If
Yeah, or skewed results that when you did it, like with your hand-picked data, it was like 96% accurate. And if somebody tried to what's it called? Like replicate your study, they would come out with like 50% accuracy. So you don't want to do that. Okay.
Thank you.
So does that answer your question, Ahmad?
Yes, thank you.
Okay, perfect. So then basically this is where I separated my training and testing data using a 25-75 train test split. And I use this function, but as I said to Ahmad, you can also create your own function because I know for some types of data, this function wouldn't work. So you can create your own, but basically just try to make it as random as possible. Over here, I am scaling my data. And as I mentioned briefly earlier, scaling your data is important when there is a large range of values. So if we scroll up, we can see that my values range anywhere from 0 to like 148 or 183. And this is a really large range of values. And this is bad for two reasons. First, it can take a really long time to train your model if the range of values is so large, so wide. And it's also really computationally expensive because like, you know that because it takes a long time. And then also it can have like a negative impact on your accuracy. So it's easier for the model if your data is scaled down and the range is smaller. So that's what I'm doing here. I'm just scaling it using the scalar.transform function for both my training and my testing data. And then I'm just printing it to make sure that it was scaled, which was. So is everyone good with that?
I'm not familiar with the standard scaling. The documentation says it's zero mean and unity standard deviation. Should you not scale the train and test parts in exactly the same way?
I did. I use the same function.
Well, so if the one that's doing the training portion only knows the mean and standard deviation of the training portion, then the test one will be potentially slightly different.
Oh yeah, I didn't think
The
of that.
random.
So I think one way to work around that would be before you split it.
Yeah,
standardize it.
do it before you split it. That would be the answer, wouldn't it? Yeah. Okay.
So yeah, maybe you guys
But
want to.
it's likely to be very small, the difference.
Yeah, or even like, or yeah.
Or maybe you transform it on the test data and then fit it separately, fit it first on the test data and the train data. I'm sorry, transform it on the train data and then fit it on the test data and the train data. See, am I making sense? So that transformation.
You measure the standard deviation and the mean on the training portion, but then
Exactly.
apply it to the test portion as well.
Yes.
Apply exactly the same transform to the test
Yeah.
portion. Yeah.
Yeah, so that would work too. So maybe you guys like want to make a note there that this would actually go before I split it. So it should actually go like here, like before this one. Does that make sense? Okay, I'm assuming it does. And then over here, I was just making sure that like this is also scaled. And then over here is where the actual machine learning happens. So all of this was just data pre-processing, like all of this is data pre-processing. And this like may seem pretty long, but let me tell you, like when your data isn't as nice as this, it's even longer. So yeah, a lot of machine learning is a ton of data pre-processing, and that's like, in my opinion, the least fun part. I don't know what like Violet thinks, but yeah. Do you have any inputs on the level of funness of data pre-processing?
Sorry, could you say that again? I was reading some other documentation.
Oh, no, I was just saying like a lot of machine learning is data pre-processing, and in my opinion, it's the least fun part. What do you think?
I think that what people talk about when they say a lot of the work is spent on data preparation, that's not quite the same as necessarily pre-processing. So yes, for a data scientist, most of your time is spent on getting hold of and preparing in some form the right kind of data, and a much smaller proportion of your time is spent on actually training your models, but it's not necessarily pre-processing as such. Pre-processing tends to be a bit more automated, but perhaps working out the right type of pre-processing also takes quite a lot of time.
Yeah. And I think I had a question from Mallory who said, do you usually select variables first or just include all of them? So I'm assuming that is applying to like these things. So you have to like pick which variables you want to go into your machine learning model, like which ones you want your model to train on. So this data set was really nice. Like I said, we didn't need to do a lot, but sometimes there's like irrelevant data. For example, I said for example twice. But if you have like the names of the people in a column over here, that really wouldn't have any effect on whether they would develop diabetes or not. So that would maybe be something that you would take out in your data preprocessing or preparation. So in this case, we did include all the variables, but if there were like variables like name or like shoe size or something, then we would have taken those out. We would have selected individually. Does that answer your question? Okay, let's see what it does. So that brings me here. This is basically where I call my machine learning classifier, where like I actually do the machine learning. And we went over this earlier, but I made my trees 100. I wouldn't recommend doing that. It took a while to train them, so I'm not going to do it again. My criteria was Gini or whatever. And you can like choose, there's like different types. Like in the presentation that I did, it was entropy. Over here, it's Gini. So you can like choose different types. There are types of criteria that are like best for different types of tasks. So I can maybe send like a Medium article or something that goes after or like goes over which ones you should choose for what task you're trying to do. And then your random state, like I said, from my experience, like trial and error, I just put 2. I don't really know like what this thing does, but yeah. And then over here, I fit it. And basically this means my model has been trained on my data. Does anyone have any questions about this?
So that what I was looking up was that criterion parameter and what the different options were, because I thought that sounded fun. So there seem to be 2 options, the entropy being the information gain, and the Gini one is that you picked is the Gini impurity. And in both cases, they're trying to do a similar thing. They're trying to see if you're gonna make some kind of split, a decision that says, you know, does it have gills or not, or you know, there are different things that you could decide. Is the weight more than 30 kilos, or BMI is more than 30 or less than 30, or you could split it on a BMI of more than 25, less than 25. Which is a better split? How do you judge it? And it's this criterion parameter that helps you to build a more useful tree by assessing how good a split is. And what you're looking for is a split so that on one side of it, you've got mostly answers of one kind, mostly the diabetics, and on the other side, you've got mostly the non-diabetics. And the cleaner you can get that separation by making just one judgment, you know, is the age more than 35 or less, or any of the other ones that you're trying out randomly. The cleaner you make that split, the better a split it is, and the more likely you are to pick it. So they're just 2 slightly different ways of assessing this purity of a split. One is information gain, and one is the impurity of a sample.
Yeah. Okay. So yeah, so basically I just fitted it there, and then over here I just created like this code basically just goes through the different I guess like testing data, and then the actual value, and it tries to like show whether the predicted value matched the actual value, and as we can see it does. But this was just like because I wanted to see what it looked like. But if you were trying to communicate your results to somebody, you wouldn't like present it like this. What you would actually do is create a confusion matrix, which is basically like, okay, I have a picture here. So basically a confusion matrix shows the percentage of like, out of all the values, the ones that you predicted were positive and that were actually positive, and those are called true positives. And the ones that you predicted were negative and that were actually negative, and those are called true negatives. So in our case, the positives are 1, like has diabetes, and the negatives are 0, does not have diabetes. So we want this diagonal line, like the numbers in this diagonal line, to be as high as possible. And we want the numbers in this diagonal line to be as low as possible. So if we scroll up here and we try to look at our confusion matrix, we can see that it did pretty well. Like our true positives are 101, our true negatives are 40, and our false positives are 34, and our false negatives, or sorry, our false positives are 17, and our false negatives are 34. So we did pretty well there, I would say. And then over here, I just calculated the accuracy, which is just one performance metric you can use. There are also so many, like there are specificity, sensitivity. I just did accuracy, which is total true positive over total true negative. And I got an accuracy of 73%, which, like I said, is pretty good for the very minimal amount of effort that we put into this. So yeah, that's basically this project in a nutshell. So this random forest classifier could classify diabetes with 73% accuracy, is my conclusion. Any questions?
So if I've understood this correctly, what we've done is we've taken a data set and we've looked at various parameters, we've tested, trained some of it, and then we performed analysis of it, and it's come out and said based on the parameters that you put in, you can predict to a 73.4375% accuracy whether or not someone will have diabetes based on that data set. Is that correct?
Yeah.
Okay, cool. Yay!
Perfect. So yeah, if nobody has any other questions, we can maybe go through the heart disease data set now. Okay, I will take that as a yes. Has anyone like run their code and got a different accuracy? No, everyone got 73%. Okay.
Let me ask a different question. Did anybody run their code and train their own classifier, or did everybody just watch this one?
I watched.
I watched also. And I must say, you didn't share the link to the data.
I did not.
Yeah, you
Yeah,
just shared
it was in the chat. It was.
Did I miss it? I don't see. I see the link for the workbook for the notebook, but I don't see the link for the data.
Okay, I can share it.
Watching was plenty enough.
Here.
9 p.m. in the
No, that's the wrong one. Sorry.
I was able to run it, and it runs okay once you've got the data in there, like you said.
Cool. What was your accuracy? I just put the data set back in the chat, by the way.
Thank you.
It was 73.91, so very near to yours.
Yeah, so a little better. That's pretty good. Okay. So yeah, let's go over the heart disease data set now. So I'm gonna close these tabs. Okay. So I already put that in the chat. Let me know if everyone can get to it. So this one's actually the one that's from UC Irvine. I apologize. The P90 was not. Okay. So it's the same deal. Basically, I download it, sign in. This computer is like older than me, so it's taking some time. Okay. And then I'm going to go to my Colab. Remember to make a copy. So file, save, oops, not, let's save it. Save a copy to drive. Oh, wow. Bridget's is very good. So in this case, we're not using random forest, we're using 90 days. Yeah, so I'm going to upload my data to my runtime. Okay, so I've uploaded it. Yeah, okay. So basically this is where I'm importing all of my libraries again that make life super easy by only having me code like 2 lines of code. So I'm just gonna run that. And if you guys are getting any errors as you're going through this notebook, it's probably because you haven't imported the libraries in the beginning. That happens to a lot of people. So just make sure if you get any errors and you can't like figure them out, it's probably this. So just go through and run it. And then over here, I'm doing the same thing I did in the last example where I'm creating a data frame. But this time my data set is called heart.csv. So that's what it's called. That's what I'm calling it as. And then if I run it, I get like this really nicely formatted table of like all these different parameters. And there are a lot of like, what is it called? Like abbreviations here. So I just wrote like a short paragraph about what each of them mean. So obviously age means age, sex means gender. 1 is for male and 0 is for female. Chest pain type is CP, so that's over here. And it ranges from 0 to 3. 0 is the least like painful and 3 is the most intense. TRUST BPs stands for resting blood pressure. Right after the person was admitted to the hospital, and that's in millimeters per mercury. COL stands for serum cholesterol level. And then FPS stands for fasting blood sugar. And if a person's fasting blood sugar is greater than 120, if yes, then they get a 1. If no, then they get a 0. REST ECG stands for resting electrocardiograph results. So over here, FAL-ACH stands for maximum heart rate achieved, which is over here. That's pretty high. XANG stands for exercise-induced angina, which is here. 0 is no, 1 is yes. And angina is basically a fancy name for chest pain. So if you get chest pain when you exercise, then that is bad apparently, and you get a 1 in this column. And then old peak stands for ST depression caused by exercise compared to ST depression caused by rest. And this is what ST depression is. So as you can see, this is a normal like graph of your heartbeat. And then this is ST depression. And apparently you do not want ST depression. So you do not want your heartbeat to look like that because this is what it should look like. So yeah. And then slope stands for the slope of the peak of the ST segment during exercise. So I'm assuming like this is the peak because these two look the same. So yeah. CA is the number of major blood vessels colored during fluoroscopy. So basically what that is it's like if like when they're trying to visualize which one of your blood vessels are blocked, the ones that get colored are the ones that allow the dye to flow through them. And if the dye is able to flow through them, that means that they're not blocked. So you want more blood vessels to be colored. If they're not colored, that means that they're blocked. And that is bad because now you're at risk for a heart attack. So yeah. FAL stands for if there was, is, or never was a heart defect. 3 means the heart was normal, 6 means the defect was fixed, and 7 means there's still a defect. So this would apply to like if you had any congenital heart defects or anything like that. Target stands for if the person has heart disease or not. So this is what we're trying to predict after that very long explanation of all the parameters. So target is what we're trying to predict. Is everyone good with that? Okay. So yeah.
Do you know, Julia, are there some people in there who don't have heart disease? Because the ones that you've listed all do.
Oh yeah, this is
There must be some, right?
Yeah, there definitely are. This is just the first 4 or the first 5 just happen to have heart disease. So, okay. So yeah, so over here I'm doing the same thing where I got the column of the outcomes over here, and I printed it to make sure it was okay. And it's very long, as you can see. So as you can see, a lot of people here actually don't have heart disease, but some people here do, and yeah. It's not like a very good randomized data set because they're all like in a line. So yeah, I just closed that because it was very long. And then over here, I am splitting my training data and my testing data using train test split. And over here, I'm doing a 20/80 train test split. So you can do 30/70, whatever you want. And then I'm also scaling my data here. And this should say so 90 days to train faster. And I look at my data and see that it is nicely standardized. And then over here, since we've already gone through one example, I'm going to give you guys the opportunity to call your model and fit it to your data in that box. And I will display the code to do that over here. But you're going to have to like fit it to your specific data. So I don't know if yours is called stats or not. So I'm going to give you guys a few minutes to do that, and then I will follow up with what I do. And maybe just like have a reaction when you finish. Like where can you do reactions? Oh, like maybe like this when you finish.
Can you go to that last part again? Because I was uploading the data set and I can't multitask.
Sorry, you sound a bit quiet. Can you repeat that?
Sorry. Yeah. Can you go through that last thing that you've asked us to do again? So I've uploaded my data because I can't multitask. So I was uploading the data set. That's all in there. I've run my libraries program. And then what did you ask us to do?
Yeah. Okay. So basically over here, just call the model and fit it to your data. So basically just like follow these instructions, but for your data.
Okay. I think.
While we're doing it, could you please explain what naive Bayes is and how it works? I know you already did, but I could not fully capture that. So could you like go over this again, how it actually works?
Yeah, maybe. Oh, okay. So do you guys like still need this, or I'm just gonna put this in the chat so you guys can have that for reference, but I'm gonna use a whiteboard to explain it because I think that might help with visualization. Okay. So actually just kidding, because apparently I can't share whiteboard here. How can I do this? Or I can annotate. Okay, I'm just gonna annotate. So can you like see if I draw this?
Now, I think like if you go to the share screen option, it has an option of like Zoom has integrated whiteboard. So if you could go.
Yeah, we can see you drawing on the.
Okay.
Yeah, that also works. Yeah. Okay.
Yeah. Yeah. I mean, this computer is like really old and the Zoom isn't updated, so that's probably why I can't do the whiteboard. Okay, but basically, in your inbox, imagine your inbox, and you have like 100 messages in your inbox, and some of them might be spam, and some of them might not be spam, but you don't know which ones are. You don't want to go through all 100 of them to like weed out which ones are spam and which ones aren't. So that's like your problem. So say two of these messages out of these 100 are spam. So 2 out of 100 are spam, but you don't know which 2 are. So if you chose like any random message from your inbox, there's a 2% chance that that message will be spam, like just randomly, if from the group of 100. But what if I told you that both of the emails that are spam have the word deal in them? So then you're like, okay, that like makes the group a lot smaller, because out of this giant inbox of 100 emails, only like 10 of them have the word deal in it. So now instead of 100, your group is only 10 emails long. And out of those 10, still only 2 are spam, because like 2 in the main inbox are spam. So you're not going to grow the number of spam. So 2 are spam. But now if you go through this group of messages with deal in it and choose 2 randomly, then there is a 20% chance that you will pick the one that has spam in it. So it basically greatly increases your probability of choosing the spam email by making the group smaller. And how it does it is it basically says, what is the probability of an email being spam given that it has the word deal in it? And by adding like that condition, given that it has the word deal in it, you make the bottom part of the fraction a lot smaller. So you increase the probability of, that's just supposed to be a 0. So you increase the probability of you choosing whatever you're trying to choose. And that's how naive Bayes works. Does that make sense?
Oh, that's cool. Thank you. That does make sense.
Okay. So did everyone get a chance to code their naive Bayes?
With an error, I've got a name error.
I was muted, sorry. Maybe do you want to share so we can go through that error and see if anyone else got it?
Hang on a minute. Let me just.
I'm going to stop sharing.
Okay. So, Isaac. I'm just going to share my screen with the class. Zach is my son, by the way. Right. Can you see what my screen now? So I imported imported the heart CSV. I went through and I ran each of these, or so I thought I did. And then when I got to the bit where it says that I thought we had to do what we had to do, I copied and pasted from, good Lord, that goes on forever. Copied and pasted model equals MB, and this is what I got.
Yeah, so it says NB model isn't defined. So if you like scroll all the way up to back when you imported everything, what did you like name?
No, it's not an importing. It's just NB model isn't instantiated. There isn't a bit that says the first line from the slide you didn't copy, Bridget. There were 2 lines in the slide that you needed to copy in.
Okay. I can't get back onto.
Actually,
they're in the chat.
I just saw one. Right, my apologies. I have to stop sharing to go back into the chat. I can't find the chat whilst I'm sharing.
Yeah, actually, Bridget, you don't need to copy the first line because it was already included in the Google Colab. So, but we just named it something different, which is why you had a naming error. So if you scroll like all the way up and underneath the imports, you see that we've named the machine learning model NB instead of NB model.
Yeah.
So that's where you're getting the name error from, because in my presentation, I named it NB model, but in the Google Colab, I named it NB. So
I should change that to model equals NB dot fit.
Yeah, because over here, our model is called NB. So down here, it should say, or, oh, I don't like have it coded, but it should say, like, yeah, model equals NB model dot fit. And then let me know if you're still getting the error.
I still got an error. But give me 2 seconds whilst I try and change it at the top. NB model equals. Run that. How do I run it all, or do I have to go through each one and run it again now?
No, if you've already run the beginning cells, then you can only run this one, or you only have to run this one.
Okay. No, I still have a problem.
It was needed. Do you want to share your screen again?
Certainly. Okay, so up here, I did this.
Okay.
Did I? Yeah,
Did you run
there.
that?
Is
Did
that
you
right?
run that? I mean, it's right, but did you run that cell?
I believe I did.
Okay.
Um,
Oh, yeah, that's true.
There are no parentheses when you're instantiating the model. You have to put parentheses as well next to the Gaussian NV, I believe.
So it like knows it's a function. Otherwise, it thinks it's available.
okay. Here I have to put.
No, like, you know how df.head has two empty parentheses that take no parameters? You have
Yes,
to do something for Gaussian NV because we're not giving it any parameters, but it's a function.
like that.
Yeah.
Haha. Right. Do I have to do all of these on the way down, or just get straight
No.
down
You can just go straight down.
to here?
Yeah. Okay. So
Have you
both-
done it? Have you done it twice? You renamed it there to NB model,
Yeah. So basically,
and then down here it's back to NB.
yeah. So name it back to NB model now because you named it NB model up there.
Right. Like this.
Yeah.
Yes.
Cool.
Thank you, everyone.
So is everyone good with creating and training your model now?
Thank you for your patience, everyone.
It was a great learning exercise, I hope. Okay, I'm hoping you guys are. So I'm gonna go back to sharing my screen. And then don't mind this, I have an error here because I didn't run the cells before this. And so my training data isn't defined, but like technically mine should also be trained. And if it were trained, then I would move on to predicting what my how my model performs on my testing data. So that's also super easy with scikit-learn. You just do predictions equals model.predict stats test. And the reason I'm doing model.predict is because my model is called model, my trained model. So yeah. And then over here, I'm creating a confusion matrix with the scores of the testing data and the actual predictions. And then you can print it here. And then over here, I have just included an image of a confusion matrix with all the different performance metrics that you can use it to measure. And over here is basically just, actually, let's just stop there. So over here, can everybody just print a confusion matrix? And in the meantime, I'm going to actually train my model. I'm going to stop sharing so you guys can print it without me telling you guys. Okay, and then you guys should have something that looks like this. Is that what you guys have? Kind of similar? Okay, so then this is the image of a confusion matrix. And you should be able to, this is like basically, it tells you how to calculate different statistics. And over here, this is accuracy. So accuracy is true positives plus true negatives over everything in the matrix. So can you, over here, calculate accuracy? Yeah. Okay, so has everyone calculated their accuracy?
I am an 8. Oh.
And then keep in mind, this is a matrix. So if you want to pull values from it, like this value would be 0, 0. This would be, I think it's rows and columns. So 0, 1, 1 or 0. Oh, wait.
I think people might struggle with the indexing because I don't think we had it in the Python refresher. I think you had some examples in the previous data set, but not
Okay.
everyone will be familiar with the indexing.
Yeah, maybe. Okay, so maybe I can go through how to index values in this matrix. So con matrix is a matrix, and I want so if I say con matrix index 0, I get this array 31 0. So that's because basically this is the 0th row. So I get this row. But if I want like this number only, then I think I would do like 0 0, I'm pretty sure. Yeah. So I would do 0 0 and I would get 31. And then it's been a while since I took algebra, so I like forgot. This would be 0 1, I think. Yeah. And then this would be 1 0. Yeah, or no. Yeah, it would be. And then this would be 1 1. 30 would be 1 1. And yeah, so that's how you pull all the values. So I would just like add all of those up, and then my true positives and true negatives would be 0 0 and 1 1. And so I would add those. So basically like that. And then in parentheses, because the order of operations, and then I would divide that by the total number of values. And then that would take a while to type, but basically I would add up all of those, and then that should give me my accuracy. So just like pausing here, we definitely did something wrong when we were training it, because we should not be getting such a high accuracy. Like over here, accuracy is 100%, because we don't have any false negatives and we don't have any false positives. And I don't think it's anything we did wrong with the training. I think it's just like this data set is bad, because like there's nothing really you could have done wrong with the training. We didn't really, you know what I mean? Like it was done automatically. So yeah, so I would just say like if you get really high accuracy values, you should be skeptical of them, especially when you get 100% accuracy, and try to like find what you did wrong.
So
yeah.
when you say that the data is bad, are you kind of leaning towards that it doesn't have enough variation in the data?
Yeah. Or like we're probably trying to predict the wrong thing. Like target might not be it. Like what we're supposed to be predicting. And then over here, if you want to view the results but like make them prettier, you can have this like colored confusion matrix, but it's basically the same thing as this, but just like a pretty color. And you can like do, so basically, what can I say here? Yeah, so this is how you do it. Use PLT, which is a Python library that allows you to graph things and make them pretty. And then you can choose different colors. And I chose cool, but say like I mistype it, it gives you a list of all the colors you can choose, and there are a ton. So if you want to like play around with that, and pick like the prettiest color, I'm pretty sure like blue is pretty. I mean, if that's like, if you guys care, it's, yeah, this is like kind of pretty. I don't know. And you want this diagonal line to be like as dark as possible in blue is what like this color scheme is called. So that, you know, like your confusion matrix is accurate, or your machine learning model is accurate. And that is pretty much it. So in this, we went through the same thing where we got our data set, we separated it into different types of values, or the input and the output. We scaled it, we split it into training and testing data sets, we fit it to our model, we trained our model, tested our model, got a confusion matrix, saw that we did something wrong probably because our confusion matrix was too good. And then just like played around with the accuracy and then presented our results using a confusion matrix. So does anyone have any questions on this?
I was just going to ask one question, if that's okay.
Yeah, of course.
I was just wondering, so if you have these two different methods, like the random forest and the naive Bayes classifiers, if you were to use them both on, like, you know, the diabetes example, which comes out on top, and how do they compare with one another in terms of the predictions?
Yeah, so I actually haven't tested both of them on the same data set, but that would be something cool to like see which one works better. And yeah, that would be like a mini science project basically, because you're trying to like measure between the two different methods. So I mean, if you try it, let me know what it turns out to be.
You're alluding there to the $64 million question of, given a data set, how do you pick which of many possible methods to use? And that's kind of the black art, depending on all sorts of things of data set size and complexity and how difficult the problem is. You would reach for a different tool out of your toolbox, but that is essentially the challenge facing any machine learning practitioner. And there is never an answer. Quite often it is, it's quite possibly one of those three, but let's try them all. And each one might need quite a lot of tuning before you get the best out of it. So that's what you spend your time doing.